Okay, thanks, Lara. So I don't see if anybody is out there, so I hope we have a live audience eager to follow, and it's a really great pleasure for me to introduce our Grand Rounds speaker, Erika Darrah from Johns Hopkins. So Erika got her PhD in immunology in 2010 from Johns Hopkins, and then stayed on there for a postdoc until 2012 in the rheumatology division. She was appointed instructor at that time, 2012, and then became an assistant professor in 2014, and is still there doing her work. I've known Erika for about that length of time, maybe 2013 or 14. Been very impressed with her work. She's published many very important papers on rheumatoid arthritis, particularly how citrullination might start, how the PADs work, how they might become immunogenic, and for example, by proteolysis by granzyme B. And she discovered a number of autoantibodies against the PADs that have a great deal of interesting biology. And so she's published all of this in a good number of papers, including several papers in arthritis and rheumatology, in Annals of Rheumatic Diseases, in PLOS One, in PNAS, and in Science Translational Medicine. So I'm looking very much forward to her presentation, and invite her to open the virtual microphone. So Erika, please.
Okay, thank you so much, Thomas. Lovely introduction, and for inviting me to speak to your group. I'm sorry I can't see you in person. Hopefully one of these years things will open back up and we'll be able to see each other in real life, and I'll have some opportunity to meet the rest of your colleagues. And so today I wanted to focus, let me share my screen here. Sorry. I wanted to focus on an aspect of the story that Thomas mentioned, which is our work on autoantibodies to the PAD enzymes and our discovery of them and what they've been able to teach us about mechanism, and some interesting clinical correlates, and we think their power potentially in translation to the clinic to help hopefully identify some interesting autoantibody subsets. And I'll explain my title in a few slides. Just want to confirm that everybody can hear me. Thomas?
I can hear you loud and clear.
Great, okay, perfect. Okay, wonderful, thank you. All right, so I have a disclosure slide. They ask that we include this. So I do have a patent that's been licensed for the use of the autoantibody groups that I'm going to tell you about, and their role, hopefully their potential to define, sorry for the phone, patient subgroups in rheumatoid arthritis. I was a previous scientific advisory board member for Padlock Therapeutics and have received past and some present support from these companies, but I will not reference any unlabeled or unapproved uses of a drug. So my general objectives for today are to first review the role of the PAD enzymes in citrullination and RA pathogenesis, and then sort of tell you about two distinct subsets of autoantibodies that we identified and their unique clinical characteristics that they're associated with and potentially their mechanistic significance. And so I broke the talk kind of into two chunks. The first is primarily clinical and really understanding the clinical significance of the autoantibodies, and then ending with a discussion of how they could be driving these unique phenotypes. And so my title is sort of borrowed from Charles Dickens' A Tale of Two Cities. Those of you who are familiar with it know that it starts with this very famous quote that says, "It was the best of times, it was the worst of times," kind of like now maybe. It was the age of wisdom, it was the age of foolishness, so on and so on. In short, the period was so far like the present period. And so I'm going to tell you a story of these two groups of autoantibodies that really represent the best of times and the worst of times for patients with rheumatoid arthritis. So just to get everybody on the same page, I know you guys know a lot about rheumatoid arthritis, but just thinking about kind of a generic scheme of how we view how patients develop this systemic autoimmune rheumatic disease, we know that there is a prolonged asymptomatic phase where patients, inherit a set of susceptibility genes like PTPN22 polymorphisms that Thomas knows very well, polymorphisms in the PAD enzymes, and certainly very specific MHC class II alleles called the shared epitope alleles, and many other SNPs that predispose patients to the development of RA. And then later in life, when they encounter a series of environmental factors, these could be things like smoking, bacterial infection, or other, these kind of factors synergize to break tolerance to a very specific set of self-proteins, which we'll talk a lot about later. This phase can last, as we know, years to even a decade, and these autoantibodies can be found in circulation long before patients ever experience their first clinical symptoms. And for reasons that are completely unknown so far, there's this period of transition where you have this amplification of the immune response, elevation in pro-inflammatory cytokines, epitope spreading with recognition of multiple kind of autoantigens that occurs in a short window about 2 years prior to the onset of clinical symptoms. And then kind of once the cat's out of the bag, you get this propagative cycle of inflammation where you have, autoantigens that are driving, antibody binding. These things are activating antigen-specific T cells, recruiting inflammatory cells to the joint. And this cycle is really propagates, as we know, kind of unchecked without therapeutic intervention. And this is the stage where the patients present in clinic with the classic features of rheumatoid arthritis, things like synovitis. If not left untreated, this can progress to really significant bone and cartilage erosion and deformity. We don't see that as much anymore, as you know, because of the great therapies we do have. But RA is not just a disease of the joints. There are extra articular tissues that can be involved, such as the skin and formation of rheumatoid nodules, vasculitis, and interstitial lung disease. And this will be something I'll be talking about later. We know that a hallmark serological finding in upwards of 70 to 80% of patients with rheumatoid arthritis are autoantibodies to citrullinated proteins. These are proteins which have been acted on by the Peptidyl Arginine Deiminase enzymes, which convert positively charged arginine residues in proteins to neutral citrulline residues in a post-translational way. And this reaction is highly dependent on calcium, and this will become important later. And so the finding that such a large proportion of RA patients make immune responses to the products of this reaction has really highlighted PAD enzymes themselves as playing an important pathogenic role in the disease. The PAD enzyme family is actually a group of 5 enzymes with really diverse tissue distribution and function. The general role of the PAD enzymes appears to be in things like tissue differentiation, epigenetic control of some genes, in particular by PAD4, which is unique among the PADs in its expression in the nucleus. The rest are primarily expressed in the cytosol, but you can see they kind of seem to have a role, really wide role across many tissues in the body, and kind of the normal physiologic roles of the PADs are just really beginning to be understood. And in rheumatoid arthritis, 2 PADs in particular have gained the most attention as probably being the primary drivers of these citrullinated autoantigen production. That's PAD2 and PAD4. So PAD2, you can see, has a pretty wide tissue distribution. It's expressed in the central nervous system, skin, really most tissues, including multiple hematopoietic cell lineages, including those that are known to infiltrate the RA joint, like neutrophils and monocytes. PAD4 is much more restricted to primarily granulocytes and monocytes. Both can be found at the scene of the crime in the RA joint. This is just some histology of 4 different patients with RA looking at PAD2 expression and PAD4 expression in the synovial tissue. And you can see the brown staining is showing the expression of these 2 PAD enzymes in the tissue itself. And you can see these PADs kind of co-locate with citrullinated proteins in the tissue. So you have both the products of the reaction, the citrullinated antigens, but you also have the PAD enzymes themselves right there driving citrullinated antigen production in the joint. PAD2 and PAD4 have also been genetically linked to the susceptibility for RA development. There have been polymorphisms in the PAD4 and the PAD2 loci that are linked as independent risk factors for RA development, primarily in Asian populations, but there's been some reports in other populations as well, and you can see they kind of are scattered throughout the genes. And so kind of with that background and sort of, you know, really focusing our attention on these two PADs, I wanted to tell you about our work looking at PADs as autoantigens themselves and how we kind of learned that the PAD enzymes themselves can also become targets of autoantibodies in distinct populations of RA. And so I'm going to start with the worst of times so we can end on a positive note. So the worst of times, these autoantibodies are to PAD4. And so it was appreciated in 2008 by 3 different groups, including ours at Johns Hopkins, led by Anthony Rosen, that the PAD4 enzyme itself was an autoantigen in RA in about 30% of patients. All 3 groups found that antibodies to PAD4 were strongly associated with ACPAs or anti-CCP antibodies. And so most patients who target, about half of the patients who target the citrullinated products also target the PAD4 enzyme. All 3 groups found that antibodies to PAD4 were associated with severe disease, the most severe disease. And so this is just a graph from the Halverson paper that shows that if you look at erosive joint disease, joint space narrowing, or the cumulative measure kind of factoring in both of those features called the Sharp score, you see this trend where patients who are negative for PAD4 antibodies have lower erosive disease, whereas patients who are positive have more erosive disease, more joint damage by these measures. And these were all comers with RA, about, again, 80% positive for anti-citrullinated protein antibodies. If we just look in the CCP positive group, we kind of see this trend maintained. It lost significance, but you can see clearly that patients with PAD4 antibodies have more erosive joint disease and joint damage than people who were negative for these antibodies. And so this is just kind of a summary so far of sort of what we know about these antibodies. Again, CCP positive patients comprise about 70 to 80% of patients with RA. About half of those patients have antibodies to PAD4, and these folks have the more severe disease. Just as an interesting note, there are, depending on the study, about 2 to 15% of patients who are CCP negative who are positive for PAD4 antibodies. It's a smaller subgroup. We don't really know if these folks are different from these folks clinically yet because the groups are small, but it's an interesting thing to note, to think about, is there additional value of measuring PAD4 antibodies even at this very early stage kind of RA diagnosis? So kind of after that study was published, we started thinking more broadly about the PAD family of enzymes and kind of asked a really simple question initially, which is, are there antibodies to other PADs? You know, we know PAD2 and PAD4 are important. Whoops, I'm going to tell you about PAD2 later. But we first just asked, because we found had found recently that PAD3 was expressed at low levels in neutrophils also. So we just asked, do patients make antibodies to the PAD3 isoenzyme? And this was with an immunoprecipitation assay that we commonly use in the lab. And what we found is that in a small kind of unselected sample of RA patients, we did find some patients who were positive for PAD3 antibodies. But when we looked at the data, we found that only patients who were positive for PAD4 ever made antibodies to PAD3. And it was a perfect correlation. There was never a patient who had PAD3 antibodies and were negative for PAD4. They always came together. And so we asked the question in a larger cohort, now separated by their PAD4 antibody status. And what we found is that in PAD4 antibody negative patients, we never really found PAD3 activity binding. But in PAD4 positive patients, we saw this clear subset of patients who also had reactivity to the PAD3 enzyme. And in data I'm not going to show for the interest of time, we actually found that these were cross-reactive antibodies. This group of antibodies was able to bind to an epitope that was shared by the PAD4 and the PAD3 enzyme. So we call these antibodies PAD3-4 cross-reactive antibodies. That's probably how I'll refer to them for the rest of the talk. And you can see that this PAD3 reactivity wasn't present in other control groups. And so now we had sort of 3 populations. We had the PAD negative group that was negative for both specificities, the group that had PAD4 antibodies but no cross-reactivity to PAD3, and then the PAD3/4 cross-reactive subset. And so we wanted to know if there were any interesting clinical differences between these three groups. And so we looked in this cohort, the ESCAPE-RA cohort that was compiled by Joan Bathon and John Giles when they were at Hopkins. Initially, this cohort was compiled to look at cardiovascular outcomes in RA, but had a huge amount of information, really detailed clinical information and longitudinal on joint disease and other aspects of the disease. And so when we looked in this group, this is a busy table, so the colors will stay the same. So PAD3/4 is in green, PAD4 only is in blue, and PAD negative is in orange. And when we compared clinical characteristics between the groups, we saw some interesting things. The first is that patients with the cross-reactive antibodies had the longest disease duration compared to the other two groups. Kind of interesting, maybe suggestive of epitope spreading from maybe recognizing the PAD4 enzyme first to the PAD3/4 cross-reactivity kind of being acquired later. We found that, not surprisingly, these cross-reactive patients had a lot of CCP positivity as well, and they were enriched for people who had these classic HLA-DRB shared epitope alleles, these MHC class 2 alleles associated with RA development, so very classic RA risk factors. And we also found this striking finding where patients with PAD3/4 cross-reactive antibodies had the most severe joint disease. Their SHARP score, their measure of bone erosion and joint space narrowing, was by far higher, median score of 49 compared to 9 and 6 in the other two groups. So we look at that graphically, we can kind of see the same trend in a crude model and an adjusted model, adjusted for these other covariates that might affect joint disease. We can see again that this PAD3/4 cross-reactive antibody subset, these folks really had the most destructive joint disease, really much more than patients who only recognized PAD4 or those that didn't recognize the PADs at all. And this held up in both models. I told you that we had longitudinal data on these folks, and so this was their baseline visit, but they were followed for a 3-year period, and we could track the change in the severity of their joint disease over time. And what we found is that patients with the PAD3/4 cross-reactive antibody not only had the most severe disease at baseline, but also were more likely to progress and continue to erode their bone at a higher rate than the patients in the other two groups. And this was despite sort of equal treatment between the groups. This was an observational study, not a treatment study, so patients just kind of were on what they were on. You can see a lot of biologic and non-biologic DMARD use, but it's not different significantly between the groups. Yet, folks with the PAD3/4 antibodies continued to progress in their disease. This kind of series of observations was confirmed in a later study in collaboration with Lou Bridges' group at the University of Alabama. This is data from his CLEAR cohorts, which are a study of African Americans with rheumatoid arthritis. And you can see a very similar picture in terms of the erosive joint disease. Patients with the PAD3/4 antibodies had a much more severe erosive joint disease burden than patients in the other two groups. And we've since seen this trend repeated in other cohorts with Ted Michaels' group and others. So it seems robust across RA cohorts, across demographics, that these antibodies really do seem to identify patients with a more erosive joint disease course. But as we know, RA isn't just about the joints, and I mentioned interstitial lung disease. The ESCAPE-RA cohort, as I mentioned, was designed to look at cardiovascular outcomes. And as part of that, all the patients in the cohort, regardless of symptoms, got high-resolution chest CT scans. And so they were also able to assess lung disease in a completely unbiased way. This lung disease was evaluated by a blinded radiologist and scored. And what we found, this study was led by John Giles, was pretty shocking and sort of unexpected. We found that not only do patients with the PAD3/4 antibodies have the most severe joint disease, they also had the highest evidence of interstitial lung disease on chest x-ray. And now most of these patients were asymptomatic, so they didn't have symptoms of ILD, but they have radiographic evidence of inflammation in the lung, as you see here, both in crude and adjusted models. And just to remind you, interstitial lung disease is the most common type of RA-associated lung disease. It's diagnosed in only about, depending on the cohort, 10 to 25% of patients, but is clinically silent in a much larger group of people, really as evidenced by this study as well. And not surprisingly, smoking, kind of an extra insult to the lung, is the biggest risk factor for RA-associated ILD. And I don't have that data here today, but when we looked to see if there was synergy between a history of smoking and PAD4 antibodies, we found a pretty dramatic increase in the evidence of a presence of ILD in patients who smoked and had PAD4 antibodies compared to the other two groups. The odds ratio was actually 60, which is kind of really high. So there's some synergistic effect potentially. We don't understand the mechanism yet between smoking and the presence of these antibodies. So just kind of to summarize everything I showed you, I already told you that PAD4 antibody positivity is found in about half of patients who have anti-citrullination protein antibodies and are associated with more severe disease. We went on then to find that about half of these folks have antibodies that cross-react with the PAD3 enzyme, and these really are the people who have the most severe destructive joint disease, but also the highest risk for ILD. And so you can see that severe disease kind of tracks with this outside group of people, and likely the severe disease observed here originally was largely driven probably by this group of people that hadn't yet been discovered. And so you can kind of see sort of like a Russian nesting doll sort of scenario developing where we can segment the RA population into smaller and smaller subgroups based on this serology. Okay, and so I want to switch gears a little and tell you kind of about the other side of the coin, the best of times, which surprisingly were people who had antibodies to the other implicated enzyme, PAD2. The rainbow is probably a bit of an exaggeration here. These people do have rheumatoid arthritis, they're still sick, but their phenotype is different and their risk factors seem to be different. And so I'll just say we didn't expect to find what I'm about to tell you. You know, we weren't necessarily surprised to find autoantibodies to PAD2 in patients with RA because just like PAD4, it's in the joint. There are SNPs associated with disease. It citrulinates known RA autoantigens. And you can see here compared to healthy controls, we had the kind of, again, our convenience sample and then our escape RA cohort, that same cohort I told you about. You can see that there's a subgroup of RA patients that have antibodies to PAD2. It was 18.5% of the escape RA cohort. And when we looked at the clinical characteristics of these folks, purely just segmenting them into PAD4 antibody positive versus PAD2 antibody positive versus PAD2 antibody negative, we saw a very different picture than we saw for PAD4. And so PAD2 antibodies were not associated with the classic RA serologies at all. You can see it's kind of a 50-50 spread. The antibodies weren't inversely associated, they weren't positively associated, there was just no association. But we did find this inverse association with the shared epitope. So this classic RA risk factor was actually less likely to be present in patients who had antibodies to PAD2. This is shown graphically here below. Patients with antibodies to PAD2 are in purple, and those who are negative are in gray. And you can see having any shared epitope, you were less likely to have that, you know, 1 allele, 2 alleles, if you had PAD2 antibodies. But interestingly, these patients were more likely to have this group of alleles that had been classified as S1 alleles by Guterud et al. This was sort of an extension of the sort of shared epitope classification where they further segmented all the HL alleles, they kind of binned them into different categories. And so classic shared epitope alleles are sort of these S2 and SP3 alleles. This is then a classic shared epitope. S1 alleles are kind of a totally different set of alleles that they showed were actually associated with milder RA, less seropositivity, sort of a less progressive course. And so this was sort of the first clue that maybe anti-PAD2 antibodies are a little bit different. They're associated genetically with this group of alleles that seem to portend less severe RA. And so we looked at that, the relationship between the joint disease and PAD2 antibodies. And so PAD2 positivity is in the solid shape. PAD2 negative patients are shown in the open shape. And you can see that patients with PAD2 antibodies at our baseline visit here had fewer swollen joints on physical exam in an unadjusted model, and then in 3 different adjusted models, adjusting for things that could affect swollen joint count. And you can see consistently PAD2 antibodies are associated with a lower mean swollen joint count at baseline. But again, this is the ESCAPE-RA cohort. We have longitudinal data. And so we looked over time, looked at that mean swollen joint count in each of 3 visits and averaged that. And you can see that just over time, these patients have less swollen joints over a 3-year period if they had PAD2 antibodies. PAD2 antibodies were not associated with any differences in erosive joint burden at baseline. So whether patients had PAD2 antibodies or not at their baseline visit, their erosive joint disease burden really wasn't different. But what was different was their progression over time. And so if we look at the annualized change in their SHARP score, the change in this score that measures joint erosion and joint space narrowing, we can see this sort of negative trend toward where patients who had higher levels of PAD2 antibodies had a smaller change in their SHARP score over time. Looked at a different way, we can also look at the frequency of radiographic progression. So how often patients are showing a worsening in their SHARP score over time. And again, you see this inverse relationship where patients with the higher levels of PAD2 antibodies actually had less evidence of radiographic progression, were less likely to progress over time. And this again was in an adjusted model for these various covariates. And again, sort of in a very different picture from what I showed you for PAD4 antibodies, patients with PAD2 antibodies were less likely to have radiographic evidence of interstitial lung disease. As you can see here, again, solid shape is PAD2 positive patients, open shape is PAD2 negative patients, and you can see this is their mean ILD score, this blinded score that was given, or the score that was given by a blinded radiologist that characterized the extent and the type of interstitial lung disease. And you can see here when the radiologist kind of binned it in terms of is the primary ILD pattern a ground glass opacification or a fibrotic phenotype, you can see that PAD2 antibody positive patients were much less likely, really none of them had evidence of ground glass opacifications on their chest CT. And this is just similar frequency of ILD, not surprisingly given this data was also less, so patients with anti-PAD2 antibodies were less likely to have evidence of ILD on their radiograph. And so I think, you can appreciate that these are really different groups of people, and there's very little overlap between the patients who have antibodies to PAD2 and the patients who have antibodies to PAD3. 4 cross-reactive antibodies. Really, there were only 3 people who had both specificities. And so we wanted to think about looking at the utility of using these 2 antibody biomarkers together, and do they give you kind of additional information or more powerful information than using either one on their own. And so we excluded these 3 people from the subsequent analysis, because we don't know which antibody may dominate and the group was so small. So we only focused on these 31 people versus these 17 people to look at the clinical effects of analyzing both antibodies together. And so when we did that, again, this is the ESCAPE-RA data. So we have 3 groups. The first group are people who were negative for both antibody types. Group 2 were those who only had PAD2 antibodies. And group 3 were those who only had the PAD3-4 cross-reactive antibodies. And what you can see when viewed this way is the groups are much more different from each other than they are from the negative patients, right? So there's some power in analyzing these 2 together in terms of predicting the risk of radiographic progression. And so again, the similar trends that we've seen, but you can see that PAD3-4 positive patients were much more likely to have a change in their SHARP score over the year, or any increase in their SHARP score over the year compared to PAD2 positive patients. And so we think there may be some clinical utility. As I said, we do have a licensed patent to a company that's aiming to make a clinical test for these two antibody subtypes for helping with RA prognosis. And so just to summarize the clinical aspects of what we've learned so far, kind of comparing the best of times and the worst of times, I told you that these patients in these two groups really differ in terms of their genetic risk. So PAD3/4 antibody positive patients are more likely to have these classic RA-HLA alleles, the shared epitope alleles, whereas PAD2 antibody positive patients are not, and instead have a different group of alleles that are associated with milder RA. They have different relationships to anti-citrullination protein antibodies, with PAD3/4 antibodies being strongly correlated, and really seeing no relationship over here with the PAD2 antibodies. Again, PAD3/4 antibody positive patients have the most erosive and progressive disease and the highest risk of ILD, whereas you see an opposite picture in patients who have PAD2 antibodies. So, you know, I'm a basic scientist, I'm an immunologist. I just have been lucky to collaborate with a lot of rheumatologists and do a lot of translational studies. But at the end of the day, I was really curious about how you could have these two antibodies. You know, generally having an autoantibody is not a good thing in any autoimmune disease, right? Tolerance is broken to self, it's not good. But somehow you have very different clinical pictures associated with these two autoantibodies. And so how could this be happening? And was there a mechanistic role for either of these antibody groups in disease? And so we first studied the PAD3/4 antibodies. We found these first, you know, these were very interesting to us. So we spent a lot of time thinking about those. I'm going to tell you about those first, and then tell you kind of what our working hypothesis is for the PAD2 antibodies. And so in thinking about how antibodies could drive disease or modulate disease severity regarding the PADs, it was important to think about what the PADs do and how they work. And so I spent a lot of time looking at the crystal structure. This is PAD4, but PAD2 is really similar, and how the enzyme gains its catalytic activity. And so it was known from the structure in 2004 that PADs are highly calcium dependent enzymes. And so when calcium is not around, this is sort of what the PAD looks like. Acidic residues are here on the surface, you can see in red. When calcium binds, you see a pretty dramatic change in conformation. Most of these acidic residues are buried, and you actually get the formation of the active site cleft. So this is where the protein goes in, gets citrulinated, and pops out. So you actually need calcium to form that cleft in the first place. The other place you see pretty dramatic conformational changes is down here. You see this formation of this kind of horn-like structure. It's actually an alpha helix that's formed with 3 calcium ions binding in this region that's, you know, absent here. So 3 calcium ions help to stabilize this alpha helix. 2 calcium ions bind up here to form the cleft. And so we started to think in terms of the PAD3/4 cross-reactive antibodies is, could they actually help to activate the PAD4 enzyme? If you're activating the PAD4 enzyme, you're generating more citrulinated protein that could be bound by anti-citrulinated protein antibodies, and you can imagine that that could provide fuel for the fire and accelerate or amplify inflammation in RA. And so we tested that hypothesis first with a really simple in vitro biochemical assay. We took purified human PAD4 in a test tube, mixed it with a known substrate, which was histone H3, in the presence of increasing amounts of calcium. And here we have no antibodies, so this is kind of the natural activity of PAD4 under this condition. And our readout was citrullination of histone H3 by Western blotting, and so the antibody will only bind, you'll only see a signal if the histone has been citrulinated. And you can see that in vitro, you really need high amounts of calcium for PAD4 to be maximally activated to citrullinate histone, and that when you titrate down the amount of calcium, you get a pretty precipitous drop-off in activity. When we purified the antibodies from the serum of patients who only recognized PAD4 but didn't have that cross-reactive ability, you see a pretty similar picture, a slight extension in the activity of PAD as you titrate down calcium. But when we added antibodies from patients who had these cross-reactive antibodies that were associated with the most severe RA, you see a really different picture. So you see maintenance of PAD4 activity over an extended range of calcium concentrations, all the way down to the lowest one we tested, which was 0.2 millimolar, really very physiologic dose. Just to remind you that the extracellular calcium concentration that's measured in serum, even in synovial fluid, is between 1 and 2 millimolar. In synovial fluid, it's actually a little bit less, or closer to 1. Intracellularly, it's really in the micromolar range. Unless a cell's activated, it can go up a little bit, but really isn't going to get much past this inside a cell. So you can see the cross-reactive antibody was really able to activate PAD4 to citrullinate substrates, really at much more sensitive to calcium, could operate under much lower levels of calcium. And so we just quantified this in multiple experiments to make sure this wasn't a one-time thing, and I graphed the results here. And this is basically showing the fold change in citrullination at each calcium concentration here versus here, or here versus here. And so you can see that the PAD3/4 cross-reactive antibodies really can amplify citrullination several hundred-fold over, no antibody alone, really at these low calcium concentrations, these sort of more physiologic calcium concentrations. You see a little amplification here as well, but it's they really shine at this lower calcium range. And so we did another series of studies, different blocking studies, enzyme kind of assays, and found that this is probably the primary area where these antibodies are working. And so Thomas mentioned that we did some work on looking at the susceptibility of PAD4 to proteolysis by the protease granzyme B. So granzyme B cleaves PAD4 at this yellow residue here, this aspartic acid 388. And when we mixed PAD4 with antibodies from these patients, the cross-reactive antibody patients, you no longer got proteolysis at this site, really suggesting that the antibody was kind of binding in this area and protecting that aspartic acid from being accessed by granzyme B. If you had antibodies from a patient who had PAD4 antibodies alone, you still got proteolysis at this site, suggesting that they were binding elsewhere and not able to protect. And so with that and some other mutagenesis studies, we kind of honed in on this area, at least, as the area that was being affected by antibody binding. And so just to remind you, this is the area that binds 3 calcium ions. And so our hypothesis with this is that the antibodies are binding to PAD4, helping to induce those structural changes that normally are achieved by calcium ion binding, but instead are achieved by antibody binding. And so these antibodies are sort of hijacking the normal function of calcium, locking PAD4 into an enzymatically active conformation. And so we kind of published this result before this was found, which it was kind of nice to see this because it helped really to further explain the hypothesis. And so this is a crystal structure of PAD2, actually. I said they were pretty similar. And this was Paul Thompson's group. And what he found when he crystallized PAD2 without calcium or with increasing amounts of calcium is, again, this formation of this alpha helix when calcium ions bound. But what he was able to show is that calcium ion binding here kind of happened last and sort of flipped the switch to lead to full activation of the enzyme. So he called this area the calcium switch. And so our, kind of language we're using now is that the antibodies bind to this region and help to flip that calcium switch, lead to full activation of the PAD enzymes, even without as much calcium around. I'll say right now we have some ongoing studies to try to crystallize the PAD4 enzyme in the presence of human monoclonal autoantibodies that bind to PAD4. These antibodies were cloned with funding in a partnership with MedImmune when Thomas was there, so that's how we originally met. And so we are now going on to try to crystallize, make co-crystals, and really map the epitopes of where these cross-reactive antibodies are binding to really understand more at the molecular level how they're helping to flip the calcium switch. And so this was a nice kind of research highlights article that was published along with our paper in Science Translational Medicine, showing kind of a summary of how these antibodies might bind. I told you they bind to PAD4, maybe help flip that calcium switch, leading to full activation of the PAD enzyme. that PAD enzyme can then citrulinate more substrates. We know that PAD4, while it's normally intracellular, in RA, it can get outside of cells and can be found floating around the synovial fluid. And so that would be a place where this interaction could happen. PAD4 could citrulinate a bunch of things extracellularly, contributing to generating targets for anti-citrullinated protein antibodies and perpetuating the inflammation in the joint. So that's a nice, tidy story, right? It was exciting. It made sense. Kind of then the antibodies can activate the enzyme, and these people do worse, and so it kind of fit together. What was a little bit less clear was how anti-PAD2 antibodies were associated with less severe symptoms. And so we started to think, well, if PAD4 antibodies can activate the enzyme, maybe PAD2 antibodies can inhibit the PAD2 enzyme, and that's how this is all working. And so we know that inhibition of PADs pharmacologically is actually helpful in rheumatoid arthritis models, in mouse models of inflammatory arthritis. And so this is an experiment that was done by Mike Holler's group, showing this is in the collagen-induced arthritis model, measuring on the y-axis here disease severity, so inflammation of the joints. And you can see in mice that were untreated or treated with vehicle alone, they developed severe inflammatory arthritis over time. But in mice that were treated with an inhibitor of the PAD enzymes called chloramidine, this is a drug that has preference for PAD4, but can also inhibit PAD2. You can see that the mice did much better. Their scores were much lower with increasing doses of this inhibitor. And this was used in a prevention model. So the drug was given first, and then the CIA was induced. But this other group here did an actual treatment model. So they induced CIA first, let the mice get a low-grade inflammation, a score of 1 in their joints, and then started to treat them with 2 different doses of a slightly different version of this inhibitor called BB-chloramidine. It's more cell permeable and has slightly more inhibitory activity against PAD2 than the regular chloramidine. And you can see it works remarkably well. At the high dose, these mice just don't get worse at all. And so there's a kind of it makes sense. There's a paradigm to suggest that inhibiting PADs can ameliorate disease in RA. So if these antibodies were to inhibit PAD2, it would be a really nice, tidy story again about how these antibodies could be associated with milder RA. And you can tell by the way I'm talking that we actually didn't see that. We saw no effect of the PAD2 antibodies on catalysis by PAD2. And so this is a busy slide, but let me just walk you through it so you get the gist here. Fibrinogen is a known substrate of PAD2. And so we used that here as our target substrate on the left side, or whole cell lysate, because maybe fibrinogen wasn't the right target, but maybe citrullination of something else could be affected by these antibodies. And so we looked at whole cell lysate just to make sure we weren't missing anything. And we see the same result in both. And so in this first lane in both figures is PAD2 mixed with fibrinogen. Here we used 1 millimolar calcium, kind of something where you could see more if you added more calcium, but you could also see less if you inhibited it. We wanted to be kind of there in the linear range of the activity. And so you can see some PAD activity is able to citrullinate fibrinogen as measured by this antibody. This is an antibody specific to citrullinated fibrinogen. If you didn't add the PAD, you don't get citrullination, so that's a good control. And then when we pre-incubated the PAD2 enzyme with these antibodies purified from either PAD2 positive sera or PAD2 negative sera, we didn't really see a change. Yeah, you can see some variation. You can see some enhancement of citrullination, probably just a non-specific effect. Maybe you see inhibition, maybe, but nothing's really convincing, and it doesn't segregate based on the presence of PAD2 antibodies. And you see a very similar trend over here. It's the same sera in the same order, same experimental design, except this is, again, hex cell lysate, not fibrinogen, and you really don't see inhibition associated with PAD2 positivity. And so, that was a bummer. Of course, you have your hypothesis. It's cool if it's right. Here it wasn't. And so we were like, oh no, how can we explain this? And then we were thinking, well, most autoantibodies, when they bind to their antigen, they're not manipulating the function of the antigen, right? That the PAD3/4 cross-reactive antibodies were sort of unique in that way, where they were able to bind and really affect the enzyme to do something different. here we thought, well, maybe the antibodies are just working like normal antibodies. They're just binding to the target and helping to clear it. And so that's sort of our working model, but I'll tell you kind of why that makes some sense. And so this paper was published by the folks at MedImmune. Again, Thomas, certainly part of this group. And what they showed is that the PAD2 enzyme was constitutively secreted from neutrophils. So neutrophils are dominant expressors of both PAD2 and PAD4, but they kind of are, the biology of the two enzymes in these cells are different. And so PAD2 in 4 different donors, when they took neutrophils and incubated them in culture, PAD2 was just continuously pumped out of those cells into the supernatant. So free PAD2 was getting outside when the neutrophil was there. PAD4 was different. It wasn't pumped out. Instead, they actually found out that it could stud the surface of the neutrophil, which kind of opens up a whole another cool set of potential mechanisms for how these antibodies could function for PAD4. But focusing on PAD2, what this could mean is if a neutrophil is coming into an area, it's pumping out PAD2 into the extracellular environment. They showed that this PAD2 was enzymatically active and could citrullinate fibrinogen. As you see here, sorry, my things are getting blocked. There we go. So again, this was in the supernatant, and they had antibodies that could actually, that they made in the lab that could inhibit PAD2 or PAD4. And so they showed that when they add an inhibitory antibody to PAD4, they didn't affect citrullination of fibrinogen, again, because PAD4 is not the one that's getting outside. But when they added antibodies to inhibit PAD2, they could inhibit citrullination, showing that PAD4 was getting out, was able to citrullinate substrates, and could be blocked by this antibody that they created that could block PAD2 activity. And so what would be the significance of that? PAD2 gets out at sites of inflammation where neutrophils are there. It can citrullinate things like fibrinogens, so what, right? Other than creating targets of ACPAs, what else could be bad about that? Well, Bill Robinson's group showed a few years ago now that citrulinated fibrinogen, and then alone, but also immune complexes of antibodies bound to citrulinated fibrinogen, could actually stimulate macrophages to secrete TNF-alpha through the dual ligation of Fc receptors and toll-like receptor 4. So the citrulinated fibrinogen was actually binding to TLR4. They ruled out LPS contamination and all the things. So it really does seem like citrulinated fibrinogen is an endogenous ligand for TLR4. And then when that's part of an immune complex, you really have this synergistic effect with Fc receptor binding that leads to kind of a really dramatic increased production of TNF. So this is buffer alone. This is citrulinated fibrinogen used to treat the macrophages. You can see an increase alone. And then when that's part of an immune complex, you see it's very significantly elevated in both of these. And so our working model for PAD2, which is a little bit harder to prove because how do you prove that the antibodies are just helping to clear the enzyme? So we're thinking about creative ways to do that. But our working model right now is that, PAD2 is getting out of neutrophils at sites of inflammation. It's citrulinating extracellular substrates like fibrinogen. Certainly that fibrinogen can be bound by anti-citrulinated protein antibodies that could propagate inflammation, but it also could ligate TLR4 itself and promote TNF secretion by macrophages. That together can lead to pretty significant inflammation at these sites and propagate inflammation at these sites. But if we have a PAD2 antibody that's not doing anything weird, it's just doing what normal antibodies do and binding to that antigen and help to clear it, neutralizing it, helping to aid its phagocytosis and removal by, monocyte macrophages, dendritic cells in the area, perhaps that's how PAD2 antibodies are helping to ameliorate disease or associated with a milder disease because they're helping to remove that PAD2 that's bad potentially to have extracellularly. So that's it. That was, I guess, an abrupt ending, but hopefully you can appreciate kind of what we've learned so far about these two antibody subgroups in terms of their clinical associations, but also in terms of their mechanistic implications for RA pathogenesis. And so with that, I will end by thanking all of my collaborators, my group who has worked on these studies. The Arthritis Center certainly have been incredible partners in having, really well-characterized cohorts. Certainly John Giles and Joe Bathon who are now at Columbia University for setting up the Escape RA cohort. It wouldn't have been possible to do any of the work I've done without having access to these incredibly characterized patients. And then all of my collaborators at different institutions and our funding sources as well. So thank you so much for your virtual attention. I appreciate it. And I'm happy to take any questions.
Thanks very much, Erika. That was really, really great. And I'm sure there's a standing ovation in each of the rooms where people are sitting right now.
Thank you.
I can see that there are some questions, so you can click on the questions and answers and tell people some interesting answers.
Absolutely. So the first is from Erika. I like the spelling. So are there anti-PAD3/4 antibodies present at time of ACPA development, or do they develop later as disease progresses? Yeah, I should have included that. I wasn't sure I was going to have enough time. So we pieced together multiple different studies that we have done in different RA groups. And so a lot of our work has been on established RA, people who've had pretty long-standing RA duration, 10 years or so. But we have done some work with earlier RA cohorts and preclinical RA cohorts with Mike Hollers. And so that study was really informative. For that study, they were patients people who were in the military who had banked serum, kind of the standard type of study now over time. And then some of them went on to develop rheumatoid arthritis, and we studied those sera from those patients who developed RA and looked at their sera samples back in time prior to the development of symptoms. Sorry, my dog. What we found there is that both ACPAs and PAD4 antibodies could develop preclinically. We know ACPAs can be found years before the onset of symptoms. We also found PAD4 antibodies. We didn't know about the PAD3/4 subset at the time, but we found PAD4 antibodies preclinically as well in about 18% of patients. And what was interesting is in most patients, ACPAs developed first, and then in a later sample, PAD4 antibodies developed. In the vast majority of patients, you saw that sequential development of ACPAs first, then PAD4 antibodies at a later date. In some patients, we didn't have an earlier sample, and so at the earliest sample, the antibodies were at the same time. And in only one patient did we find PAD4 antibodies first and then ACPAs. So the model that we've developed, that we're thinking about, seems to be that ACPAs developed first, maybe as a result of epitope spreading, anti-PAD4 antibodies develop. And then we did go back and look at that data for PAD3/4, and we only found preclinical PAD3/4 antibodies in one person, and that developed much later, right before diagnosis. So there does seem to be some evidence of evolution where the antibodies develop over time, maybe as a result of epitope spreading from the substrate to the enzyme. And certainly this associated with disease duration, patients can also have established RA and acquire PAD3/4 positivity during their established disease. So there does seem to be this continual exposure to antigen and evolution of the antibody response. Does that answer your question? Maybe, okay. Great. So then we have our anti-PAD antibodies directed against citrulinated residues on the PAD enzymes. If so, are there detriments of their autocitrulination activity the same? Okay, yeah, so we actually just published this. It got accepted by ANR. I don't think it's available as an EPUB yet, but probably in the next week or so. So we did look at the ability of anti-PAD4 and anti-PAD2 antibodies to bind to the native kind of unmodified PAD antigen or the autocitrulinated PAD antigen, because PADs do have the capability of modifying themselves, of citrulinating themselves. And what we found is the antibodies largely didn't care. They didn't care if the antibodies were native. They didn't care if they were citrulinated. They bound irrespective of citrullination status for most sera. We found a couple sera, I mean like 5% or less, that seemed to either prefer the native or the citrulinated form, but still had reactivity to both. It was just stronger toward one or the other. But the vast majority of sera seemed to bind to epitopes that were maintained, that were the same, whether it was a native or citrulinated PAD. Can you block anti-PAD antibodies with a peptide and use that therapeutically? So that was Dr. Alcon. That's a very cool idea. I think once we understand more about the epitopes and where these antibodies are actually binding to PAD4, the contact residues, and sort of how they do this job, I think we could be able to think about something like that. From what we know so far, the antibody seems to be conformational, so it's not a linear epitope, it's probably highly conformational, and so that might be a little bit more challenging to make a blocking peptide, but maybe even things like a sort of plasmapheresis in an antigen-specific way, or other strategies I think would be interesting to think about. Erika, again, are anti-PAD2 versus anti-PAD3 for patient populations associated with different types of synovial pathology? That's a really interesting question. We don't know. We haven't looked. At Hopkins, we're just starting to ramp up on kind of synovial biopsy type of studies. So in-house, that's hard to do. I think it's a really interesting question. If we can, get the right collaboration, I would love to know that. I think it's important to understand what the synovium looks like in these patients, or the synovial fluid at the least. So I think it's an important question. And thank you for the compliment. Do we know anything about the B cells that produce these antibodies? A little bit. So we did this study with MedImmune and my collaborator Felipe Andrade at Hopkins, where we isolated PAD4 specific B cells by flow cytometry and then cloned their antibody BCRs, and we're able to make monoclonal human anti-PAD4 autoantibodies. These antibodies some of them were PAD3-4 cross-reactive. Even without that, they were actually able to activate PAD4 function, and so they're the ones that we're using to make crystals. So what Felipe did to tell us a little bit about the antibodies is he took those antibodies and reverted the somatic hypermutations back to germline, so the germline V, D, and J sequences for the heavy and the light chain. And what he showed is that for some of them, even with their germline reverted sequences, they were still able to bind to PAD4. They weren't polyspecific. They didn't bind to citrulinated antigens. They didn't bind to other, highly charged things. And so for those, it seemed like these B cells were sort of born to be PAD4 reactive from birth, from the bone marrow, that they were PAD reactive precursors that acquired, affinity matured and acquired hypermutations that allowed them to interact stronger and activate the enzyme. So the ability of the antibody to activate the enzyme was dependent on the somatic hypermutations, but not the ability to recognize it. So that's all we know so far. I think there's a lot more to learn. I think I have a couple more minutes. So from Han, what is the prevalence of anti-PAD2 among CCP negative RA patients? Does anti-PAD2 have a diagnostic value in this population potentially? I think so. I'm trying to think. PAD2 antibodies were found in about 18.5% of RA patients total, and about half of those patients were CCP negative. So what's that, 5 to 10% of the CCP negative group have PAD2 antibodies. So I think there is some potential utility in maybe helping to diagnose the seronegative population. That needs a lot more work, but that's something that our industry partners are trying to do in terms of thinking about rolling this out as a clinical test. And then, is there any functional significance of the antibodies also targeting PAD3? That's an interesting question. PAD3, we tried a little bit of this. It looks like we can get them to activate PAD3. The problem is the actual physiologic substrates of PAD3 are a little less clear. Its primary role that's been studied is in the hair follicle, and it citrulinates this protein. I'm going to mispronounce its name, so I'm not even going to try, but it's a very large protein that's expressed in the hair follicle, and we haven't played around, to see if the antibodies affect the ability of PAD3 to target a citrullinate, target a bona fide substrate. But I'm not sure of the relevance of the PAD3 cross-reactivity, other than it helped us to identify a really interesting subgroup of patients, because PAD3 is really absent from the RA joints, again, primarily expressed in the skin, particularly in the hair follicle. And so I don't think of alopecia, for example, as being a major feature of rheumatoid arthritis, but I think it's worth, we shouldn't overlook it, but that's all we've done so far. Do PAD3-4 antibodies influence severity of RA-ILD? the pattern, UIP versus NSIP. The study was small, and so we haven't really looked at that level. That information is there. In the paper, it's sort of described a little bit, and I honestly, I'm sorry, I can't recall, but there was some description, but I think the numbers are too small. We really need to repeat this and validate this, I think, in a larger cohort, separate cohort, and that's an area that we're actively trying to do right now. And so hopefully we'll have more information about that soon.
So Erika,
yeah.
I think we probably need to let you off the hook. It's
okay.
past 10 o'clock already, but thanks very, very much for great presentation and great answers to all these questions. And I would invite everybody who has more questions to email Erika directly, and she has promised to reply to you.
Yes,
So
I would love to.
Thanks very much. Take care.
Thank you so much for having me. Bye-bye.
Bye-bye.
Thank you, Dr. Darrah.
Thank you.